welcome to our Gates Institute seminar. And we are thrilled to have you all here today to hear from one of our 2022 Gates Grubstake awardees, Nick Jacobson. And we are particularly pleased to have with us Aoife Cullen, who is at CU Innovations, and she is going to introduce him. So if it's okay with you, Aoife, why don't you go ahead and start?
Sure. Thanks very much. Hi, everybody. It's my pleasure to introduce Nicholas Jacobson to the seminar today. So a little bit of his background. So Nick is a CU Anschutz research faculty member and a translational clinical designer at InWorks. He received his bachelor's in architecture from the University of Wisconsin and has a master's in design from Harvard. And given his education and experience, his fascinating background with a unique path to the Anschutz campus, he's designed buildings for the aerospace industry, concert halls, ultra high-end residential buildings, operating rooms, and structures in extreme environments. And he's translated his architectural skills now and his techniques there to develop solutions to complex problems in medicine. His work focuses on 3 areas of patient-specific development: diagnostics, prosthetics, and implants. And his position combines innovation and collaboration. He works with surgeons, radiologists, engineers, computer scientists, and life science researchers to conduct translational development of new products. And his work and research has been published in books, scholarly journals, magazines, newspapers. He's lectured at Harvard, Stanford, University of North Carolina, and the University of Denver, and for companies like Autodesk, Hadid Architects, Medtronic, Edwards Life Sciences, Thornton Tomasetti, Stratasys, and Trimble. And so I'm really looking forward to hearing Nick present on his role at Anschutz and learning more about the clinical impact of his research and development. And I hope there are many new collaborations and innovations that can result from these interactions with all of you listening in today. And Nick has also kindly said that he's happy to be interrupted along the presentation with any questions. So it might be handy for you to use the reaction function to raise your hand, but we'll see how it goes in terms of the mayhem that ensues. But Nick said he's comfortable with it. So over to you, Nick.
Thank you. I need to tone down the introduction because lower expectations. So thank you, first of all, for having us. I should say with that background that I sort of know what I'm doing at any given time, but for the most part, I have no idea what I'm doing. So I have a really big team that is a part of this project and all of this projects, all of the projects that I work on. Sitting beside me here is the great Dr. Mitch Fedepo. And Mitch is working in the Jeff Jacob lab as a postdoc and has been part of the project all throughout, and is going to be able to talk a lot more about the regenerative medicine side of this project. My job is really on the computational side and coming up with the crazy ideas that make Mitch and shake his head and go, I'm not so sure this is a good idea. But I also want to say thank you for the Grubstake Award. We came out to this campus with no projects. The idea of working on 3 different levels of patient-specific models, being able to replicate the body better than anything else that's out there, then move on to prosthetics, these models that would touch the body, but not go into the body. And then hoping to be able to get funding for something that would then go into the body as an implant. And the Gates was the first one to support that work as an implant, and now we're funded on all 3 levels. So thank you for helping make my dream come true. And with that, I'm going to get into it.
Awesome. So I'm going to start us off here. Just a quick crash course on heart valves here. They ensure unidirectional blood flow throughout the heart and the body. There's 4 main valves within the heart. That's the aortic, tricuspid, pulmonary, and mitral valve. And unfortunately, these valves can become diseased either through congenital valve disease or acquired heart valve disease, and then necessitate valve replacement. So traditionally, that's been done with open heart surgery, where the surgeon would crack the heart. It's quite traumatic. More recently, the recent being the last 20 years or so, transcatheter valve replacement has become more popular, where now it's a minimally invasive surgery. A catheter is going up through the femoral artery. A stented valve is on a balloon, and they're able to blow up that balloon in place and replace the valve that way. The congenital heart valve disease primarily affects the semilunar valves, the pulmonary and the aortic, and we're focusing mostly on the pulmonary valve today as we're talking about this. And that's because the need for a congenital heart valve replacement is much greater than number-wise, and so numbers, translate to dollars, but the numbers-wise, there's much greater acquired heart valve disease, but the clinical need is really in a better pediatric valve replacement. They suffer a whole lot more. They need replacement much more often. And so that's where our initial focus is. And that's so broadly, big picture, the objective of this project was looking at transcatheter valve replacement for all. And I'll dig into this a little bit more in the next couple slides. But really what we're looking at is a patient-specific heart valve could eliminate the need for open heart surgery and really revolutionize the treatment of structural heart disease. What do I mean when I say transcatheter valve replacement for all? Well, unfortunately, these currently available valves that are made right now, they don't fit the unique patient anatomies that these patients have, right? So they're all the currently available valves, these perfectly round cylinders, and they may not fit the unique anatomies. And what that leads to is only about 30% of the current transcatheter or current heart valve replacements in the pulmonary position are able to go transcatheter. That has a much shorter hospital stay, a much lower cost, but unfortunately the majority of these are still being done surgically. So the goal would really be to have all patients be able to undergo this transcatheter. As I mentioned, this one size does not fit all, and that's really the primary objective that we're trying to overcome, but there's a lot of other room for improvement. So the materials that these current valves are made out of are also quite poor, and they have this limited lifespan. They only last 7 to 10 years and then need replaced anyways. And so there's just a lot of room to improve on all aspects of this project. And then we're going to shift gears a little bit. I'm passing it back to Nick. We'll do this game several times.
So at the base of our crazy idea is a technology that we call voxel printing. It's some projects come out of different places. And this project really came out of, it was technology driven. We had this ability, we patented here, this ability to 3D voxel print, a form of 3D printing that prints directly from medical images in high resolution without any loss of data. We've got control down to 15 micron. We can overcome a lot of limitations that currently exist with 3D printing. Exploring this technology led us to try to figure out, where does it go? Where does it belong? And through different talks like this on campus, some people have come forward and said, oh, I know exactly where this belongs. And over the course of time, we've applied this in many different areas. It was Dr. Gareth Morgan who came forward and said, I think there's this idea for a patient-specific heart valve. But to give you a basis of what this technology means, I've got a little video that I'm going to show and explain to you how voxel printing works. Basically, we use a voxel modeling software. It's different than any other software that exists. It's kind of like your computer screen, but in 3D. Instead of pixels, it's a 3-dimensional pixel. And we control every single one of these, as opposed to working on a surface. So we're able to control every single droplet, every single pixel, just like a computer screen, but in 3 dimensions. So it allows us this unprecedented accuracy for modeling and getting down to, like I said, about 15 micron, where we can control color, material property, and location. And just to have fun with it, I'm going to show you a little example of what this means with a Cheez-It, one of my favorite snacks. We 3D scanned a Cheez-It and just a basic 3D scanner and tried to recreate this using voxel printing. So you see the texture, you see the color, delicious bits of salt. And here's a recreation of it using all, yellow is really a combination of a number of different colors. Here you see the recreation in the computer. So a lot of data is being processed and done really quickly. So you see the actual on the right and our 3D printed version on the left. There's some color correction. We call it the greasy Cheez-It. But that gives you a little bit of an understanding of how this technology works. So when we apply this then to the human body, getting away from Cheez-Its, what we can show is this direct translation from a medical image to a 3D printed model, where we're able to control, basically translate information that we're getting, in this case, density-based information from an MRI. And we map that then to various different colors. And through exploring and filtering our colors, we're able to pull out more information and finite levels of detail and directly translate what we're getting into a 3D object volumetrically as well. So we're able to get these gradients, which has been really critical. The work that we did to get started along this process was just featured on the cover of 3D Printing and Ad Manufacturing, which is nice to get some recognition in the ad. So when we apply this then to the heart, here's a little cross section through the heart, and you're able to see us applying different materials to different densities of the heart. So we can be able to recreate the heart with mechanical properties. So the clear parts are, fatty tissue that's really soft. And we've got some other parts that are red, the muscle that's a little bit more stiffer. And this is just kind of a proof of concept to see if it could even be done. But it's a lead into where we're going with this. And the other thing about voxel printing is we can start to control form in really high resolution. So here, we're able to take a model and apply a microstructure to it. Microstructure that we create. And we can do this in ultra high resolution. See a grid structure, kind of this crate that is working holistically, like a kernel-based filter that works all the way throughout and conforms to the pattern or the model that you give it. We can control the color, we can control what material is being placed where. So we're able to create voids, solids, material properties. And putting these things together has really led us to this project. And I'm gonna, you wanna do?
Sure.
I'm gonna let Mitch take it from here.
Yeah, so this led to the first, or not the first, this led to kind of these three themes of what we wanted to accomplish with this project. And so number one is that patient-specific valve, like I talked about here, and it's actually,
Hey, Mitch, I just want you to let you know, with both of you, the sound is going in and out a couple times, so we just couldn't hear what you said. Okay,
Oh, okay.
Okay.
Yeah, maybe I just need to speak up a little bit. I'll try that. But again, this led to three themes for this project. It starts with creating these patient-specific valves, like I talked about. But from these patient-specific valves, like I said, there's a room to improve in terms of materials here. And so we want to make these out of these soft 3D printable materials, and that can be fine-tuning the mechanical features using that technology Nick was talking about, right? Maybe blending different materials. But part of that is this new technology, new 3D printable materials that are coming out. And I'll touch on that a bit more in a second. And then lastly, really utilizing that voxel printing technology. Can we actually include microstructures within our valve to potentially influence cell behavior? That led to three aims within the project here, and we're going to walk through our progress that we've made within each one of these aims. So we're starting with aim one here, and that's testing the compatibility of these 3D printable materials. So we started just by simply testing the compatibility of commercially available 3D printable materials. And I have compatibility in quotes here because biocompatibility can be a bit subjective. For example, we tested three different materials here: Formlabs, Med610, and Vero. They're all used in medical applications, but Med610 in particular is approved by the FDA for 30-day mucosal contact. They're biocompatible by that sense. However, the biocompatibility that we're looking for is long-term implantation, hemocompatibility as well, because it needs to be in contact with the blood the entire time. So we wanted to start just by comparing these quick three materials and get a good understanding for the assays we'll be doing, et cetera. And then we'll move on to some newer materials. So again, this is a quick, we cultured cells on these materials. In this case, we're using fibroblasts, compared it to tissue culture blank. and measured the cell density and viability and found that there were some differences, but Med 610 performed well, which we expected. And we also performed some hemocompatibility testing, which isn't shown here because these materials were, again, just kind of a trial run, but the hemocompatibility testing turned out really well. And I wanted to shout out Andrea Lauren was a master's student that was helping with this project early on. It really kickstarted this part of the project and really made some of these experiments happen. We've then been in collaboration with Dr. Jeff Stansberry. He is in the School of Dental Medicine, developing some of these novel materials. He's a material scientist and is creating some new 3D printable photopolymers for dental applications. But he claims, man, I can go really soft and really durable with some of these materials to the point that we can have potentially tissues that might mimic a heart valve, some of these soft tissue applications. So we've been working with him and getting our hands on some of these softer materials. Unfortunately, due to some staffing stuff on his side, it's been a little bit difficult, but we've gotten our hands on a couple materials so far. And that being DDI HEMA and MUDMA HEMA. I'm not a material scientist. I won't be able to tell you much more than that about what's actually in these materials or the formulations of them. But again, we did a cytocompatibility test here, fibroblast density and viability and HUVEC density and viability. Fibroblasts they're commonly occurring throughout the entire body, and they're just a good support cell. And then HUVECs, or vascular endothelial cells, would be cells that contact the entire blood surface. And they're the ones we're really focused on. And I'll mention that a little bit later in the future here. But when we looked at those compared to a negative control using 5% DMSO and then our positive control of Med 610, we found that the fibroblasts did just fine. The HUVEC viability was great as well, but actually the new materials had very low HUVEC density and about comparable to our negative control. What that probably means is we're actually seeing low proliferation of the HUVECs, which was quite interesting. It might actually be worth further exploring, but both of these materials were still too stiff for our application. They might be used in some kind of blended purpose in the future, but they're about the stiffness of your fingernails. So they had some give to them. They could bend. They were quite elastic. They weren't brittle like most other 3D printable materials here, but we need to go much softer. So we're working on getting some softer materials with Dr. Stansberry here. We're looking at the 10 to 100 kilopascal range to continue further testing. So I'm going to shift gears again here, and we're going to talk about the endothelial cell adhesion with these microstructural features. So why microstructures and why endothelial cells? Well, our endothelial cells line all of our blood vessels and all blood contacting surfaces, including heart. And they provide this hemocompatible layer between your blood and tissue. And they also then are involved in thrombosis and injury signaling. But really, it's that hemocompatible layer that we're really interested in. There's been a lot of work by other researchers looking at getting that hemocompatible layer onto other cardiovascular constructs, whether that's prosthetic heart valves or stents, et cetera, because it can provide that hemocompatible layer, prevent blood clotting, and help the construct, survive in the body there. And so what we really want to do, and this is the regenerative medicine really aspect of this project, is how can we get these endothelial cells to cover and grow over our construct and provide this hemocompatible layer. And we know that a cell's behavior can be influenced by its microenvironment through different processes such as mechanotransduction and others. And other researchers, other work has shown that microstructures on the surface or micropatterning can influence cell behavior, including endothelial cell adhesion. There's been some work looking at different pillars and pores, which I'll explain further, but one paper that I really want to highlight here, this came out of the bioengineering department here on campus. Tina was she was a graduate student that finished up about a year before I got here, and she was working in Dr. Shondis's lab and found that these microgrooves on shape memory polymer surfaces used for stents can actually increase endothelial cell adhesion. So we wanted to do something similar, but not with shape memory polymers, but these 3D implantable materials. 3D printable materials. Like I said, different groups had shown that micropores and micropillars can have an effect, and they're very simple structures to print. So we started, let's just print these micropillars, micropores. And we just went straight at it with the printer, took it to scanning electron microscopy to see how it turned out, and it didn't really work. As you can see there, the Vero print had very little microstructures show up. Med 610 turned out slightly better. The pored surfaces, though, were almost not, there's no features there. And then even the pillared surfaces, it's more like rounded hills than these micro pillars like we expected. We were hoping to print 30 micron pillars, and what we got was probably 200-ish micron rounded hills. And what that is it's a lag time in between when the droplets are actually extruded by the printer and when they're cured by the UV light. And so there's some tuning that could be done on the printer side of things, but there's also things that we could do on the design side of these things. How are we designing these microstructures to account for this? And then when it's printed, maybe they will, become the shape that we're hoping for. I just want to show off some more of these SEM images. I just love SEM and the resolution you can get out of it. So we took a step back and just wanted to say, okay, what is the minimum resolution that we can print at? We made a swath here. The left side, those numbers on the left are the height of our different features. The row along the bottom there is the diameter of our either pillars or pores, going from 15 to 300. We printed that in Med 610 with either pillars or pores again. And I boxed here in the red what I consider to be our kind of like minimal pattern that was able to show up here. So that was at 50 and 100 with these pillars. It's worth noting that smaller diameters, as we got to the 15 and 30 diameter, we did see a really interesting pattern emerge, but it was an irregular pattern. And so the printer was actually it was limited by its XY resolution in that case. And it wasn't until we hit about 100 micron diameter that the XY resolution was actually able to match what we were trying to print. But that doesn't exclude that 15 micron diameter. Again, those are things that we can, now knowing about, we can include into our design considerations. But this is our, our regular repeatable pattern here with the pores. Again, that we were at kind of that maximum diameter, and even then they're just kind of divoted wells. They're not really pores like we were hoping to see. So we reprinted these in support material. So we actually printed the whole thing upside down and then encased in support material to try to give those microstructures a little bit more support here. And what we actually saw is that the pillars performed worse. I believe it's because the support material was then, as we were taking that off, maybe affecting some of the micropores, or the support material could be, blocking some of the pillars from sitting down. Either way, found that the pillars were less well formed when printed with support material surrounding them, but the pore microstructures did. So we then seeded cells onto these different microstructures here. We again did endothelial cells and fibroblasts on pillar pores, 48 hours, did a simple live dead stain. And I again want to acknowledge Albert Mungia and Ann Wynn, they're two undergrads in the bioengineering program that really helped with the analysis of this quite recently, and just wanted to acknowledge their help. And what we found is very clearly we can actually affect the endothelial cell behavior with these microstructures. So the left is our IF images. The green is indicating our live cells. Red would be dead, and then the blue nuclei, or blue would be either nuclei or there's some background staining there. What we saw looking at the fibroblast and endothelial cell density is all of the groups with pores had very low density. So the cells really just did not adhere or did not proliferate on those groups. So we can influence cell behavior, which was very encouraging. However, if you note, the pillared groups performed just as similar as the blank group, right? So we actually weren't increasing endothelial cell behavior here, but we were able to influence it by decreasing it in the pored group. So slightly encouraging results. What this means is it's, back to the drawing board, and what it is, it's time to fine tune what that microstructure is that can actually be used to increase endothelial cell adhesion. So around the same time, we got a new printer at Inworks here, printed a much higher resolution. And so we began playing with this and seeing, you know, what other kind of prints can we make. Here's just a graded gradation, a pretty dense packed microstructures at the bottom, and it gets looser up top. We're able to print now at maybe 100-ish microns, so a little bit finer resolution there in terms of the size of each droplet. And then our XY resolution is quite good and much better. As you can see, each of those little droplets is stacked right on top of each other. We then printed just a swath of different microstructures with a slight gradation across them, just one of each really, and put cells on them just to see what would happen. So there's no real stats or data here for me to talk about, but we really wanted to just see what happened. The bottom right two images, actually, you can see very little cell adhesion. There's some green dots in there, but all those cells are floating, and you can tell they're not adherent. So those groups actually look like the cells did not adhere, but all the other groups seem to do okay. And so get there. Just want to show you guys some more of these great pictures. One more zoomed in. So really next, and this might be a good time to talk about this, but next steps are, as you can tell here, everything is printed mostly on flat surfaces with a slightly raised or lowered surface. We're printing in 2.5 dimensions. And so what we want to really start doing is start printing in 3 dimensions and printing some of these complex structures like a patient-specific, you know, organ with some of these microstructures and pushing the limits of what this voxel printing can do. So I'm going to pass it off to Nick and let him show off some of these cool prints.
One of the things I love about working with Mitch is we make a really good team, because I usually just say, let's just do it. Let's just go forward and let's try something. And then Mitch helps keep me, you know, sometimes that works. We do something like just print the pillars and see where it goes, and we learn something from it. And other times Mitch really helps to keep things rigorous. But between the two of us, we've been able to move forward. So I'm going to describe a little bit about our process now, applying all of this work back to the heart valve, now that we've got some data, some promising data, and where we want to go, and how we're going to move forward with all of this. We started this project. I love this project because there's so many people involved. I don't know how many we've listed so far. I'm going to keep listing people because it's really connected. We have a really wonderful advanced imaging lab here on campus, run by Dr. Alex Barker over at Children's Hospital. They focus on advanced imaging, and he's been figuring out how to be able to acquire a full cardiac cycle over 21 frames. Previous to this, you might be able to get systole and diastole. Maybe you can get a few in between, but we're able to now register the heart in one spot, which is a hard thing to do because you breathe, right? Your heart moves in different places. But to be able to register the heart in one location and yet still get it to show like it, image in the same location over 21 frames. So that way we're able to get a full cardiac cycle. This is the point where you're going to see one of my great summer intern from last summer, Inky, start to cringe, because this is the part where she helped us a lot last summer. She took a lot of these images and did the segmentation work on this. Maybe a little scarred still from this, but it was very, very helpful. I can't wait to show you what we've done with it, Inky. So here are the images. We first started doing this. The first thing that we did with these images is we used them in the cath lab, and the cath lab is the ultimate home for this project. But it's going to show you a couple things. One is going to show you what it means to be able to have a 3-dimensional cardiac cycle. And 2, it's going to show you the current method for how we place valves and some of the issues. This is VR, and it is something that we're using in the cath lab now for most cases. We were able to get the cardiac cine, the 30 frame or 21 frames animated, and then take a stent and start to place this in the right location to understand. Currently, they're looking at systole and diastole and measuring with a measurement tool to understand where the widest point is. So simply having this in the cath lab is revolutionary. Being able to place it, understand if it is the right size, if it's too small, too big, in the right location. It's still very crude. which I hope gives you a little sense of why what we're doing is important. That gives you a sense of what this 21 frame image sequence looks like. What Inky was able to do is take each one of those patients, we've got 30 of them. So if you do the math, 30 times 21, 600 segmentations. We're working on figuring out how to automate the process. But what Inky was able to do was isolate just the pulmonary outflow tract from each one of these frames and each one of these patients. A tremendous amount of work. But we wanted to be able to understand where the heart was moving throughout the cycle. And if we had that, then as a model, we could start to analyze that. Ultimately, the design of our valve is going to be the widest point throughout the cardiac frame or cardiac cycle at every given point. That makes sense. So what we've done is once we've taken all of the segmented pulmonary outflow tracts, we've combined them. So you have one monster pulmonary outflow tract, but really it's just like you're taking a shrink wrap at its widest point. So that way our device is actually going to be where the outflow tract is, its widest point at every point of the cycle. Because what we're looking for is a device that fits in all portions of the cardiac cycle and doesn't leak at all, isn't too small, isn't too big. So through this work, we're able to perfectly size this to the actual patient. Here's a version of a model I'm holding up. Those are Inky's fingers here with a beautiful nail polish. Sorry. Some more images of what this looks like. Now that we have all of those images, we're able to analyze these a little bit more. So it's one thing to be able to have a device that is shows the right size, but now we need to understand what how the tissues are behaving. So what you're going to see here is a morphological study of each of the patients that we've worked on. Seeing how the pulmonary outflow tract moves throughout this sequence, we're also understanding it's color-coded based on the velocity that it's moving. So this tells us a little bit about the compliance of the tissue. Higher compliant tissue is moving a lot faster. Now, patients with tetralogy of Fallot have a very unusual outflow tract. So they have a very unusual heartbeat or morphological form to this. Once we understand how that heartbeat works, what the patterns of how it's moving, where it's moving, and what velocity, we can start to apply the right material in the right place to be able to counteract these forces and take into account how the heart is beating. So here's the first example of what we've done with this. Inky was able to analyze this data and put, there's many different off-ramps to this project. It's funny, we started off trying to make a 3D printed implantable heart valve, and what we found is that there's all kinds of things along the way that we can help. This is a short-term solution, understanding where the widest point is of a cylinder, which is how they're currently placing these devices. So if we were to take this and give this to our surgeons right now, which we're working to do, we can understand where exactly to place the device at the widest point, so they don't have to just use a two-dimensional measurement tool. This shows a cylinder moving all throughout. That makes sense. Really exciting stuff, and immediately applicable. Now we're mining that data to be able to understand where the absolute maximum and minimum is, not just as a cylinder, to be able to account for how they're currently doing things, but for our device now going forward. This tells us a lot about tissue compliance, the behavior of the tissue. So we've been working with a computer science student, Gunnar and Sarah, on how many different groups are involved in this project: computer science, bioengineering, advanced imaging, cellular biology, polymer chemists. It's really a Noah's Ark of people. And so what we're doing now is we're trying to understand where the widest point is, the farthest point away from the center of this device and where the nearest point is, which is telling us exactly, where the point of highest compliance and the least compliance in this pulmonary outflow track is. We've been mapping this and trying to understand if there's a pattern. The real thing that we're looking for here is there a pattern to tissue compliance or the way that the heart beats, the form of the heart, how it's beating, with the different forms of pulmonary, with tetralogy of Fallow. We know that there are known morphological patterns for tetralogy of Fallow, and within that, we're trying to describe it even more to understand more about the tissue and describe it. Our next steps are to work in 4 different categories. So if I break this down for you, the Gates project was able to help us understand materials and dip our toes into material development. We've been able to explore a number of different materials and get started with exploring the materials here on campus. And I just want to take one note and talk about Jeff Stansberry for a minute. We've had the idea for this project for a long time, but we didn't have the materials, and that's been the limiting factor. And I've been looking up and down all around for materials. I went to the International Polymers Conference, trying to find people that could do this material. And turns out there's a guy in the building right next to me that's capable of doing this, which is really exciting. We were able to explore with the Gates the tissue engineering component, understanding how cells grow, the biocompatibility, hemocompatibility of some of these initial materials, and then the software development, which we just showed. We've been applying for AB Nexus with Rob McCurdy on the Boulder campus and Jeff Stansberry to incorporate changes to the 3D printer that needs to be made to account for new materials. So we're looking at developing new materials. The 3D printer is limited by the materials that are allowed to go through it. And some modifications, some hacks need to be made to the printer. Great Rob McCurdy up in Boulder is really great at hacking these printers. We've been working with him to try to understand how to account for this next phase of materials that come through. And then finally, we've been looking at larger funding, which brings all of these things together. So we want to be able to have the full circle of development from a new material that Jeff is able to make to what we can do and design with it, to the impact that it has on cellular growth and how we can then print it. All of these things need to start working in concert and influencing each other. We're really close to that point, and we're hoping with future funding to be able to do that. I hope I'm not missing anybody here, but I'm sure short amount of time that we've been working on this project. Wanted to mention the team at large. Again, there's Mitch, myself. Didn't introduce at the beginning Dr. Gareth Morgan, who's the director of interventional cardiology, and a guy who entertains all of my crazy ideas and puts fuel to the fire. He's been really great at helping direct us on the medical side and the application because he sees these problems every day. Jeff Stansberry, the great Jeff Jacobson, and Rob McCurdy are integral parts of the team and will become even more integral going forward. And then throughout the process, we've had a diverse group of students. Anne Mugen, Albert Mungia, Andrea Lauren, bioengineering students. Robert Elliott, who is an InWorks student and a bioengineering student that's now getting his PhD here. Gunnar Insero, computer science student who's been working in our lab. Great, Enki. Max Simon, who is now a student up at CSU, who actually had tetralogy of Fallow and wanted to come work with us after I presented at the Gates last year. Came up and said, hey, I want to be a cardiologist. I see what you're doing. I had this and I want to see if I can help. Been great. Hopefully we get him back this summer. And Rob Ducey, my collaborator at Leica Stop Motion Animation Studio. We collaborate with such weird people, but Leica has been a collaborator for a long time. They bring technology typically used for animation and special effects to be able to help solve some of these complicated projects. Although they never think about their work outside of the animation, when I start to ask questions like, hey, here's a problem I'm trying to solve, they go, oh, we do that when we try to convert a smile to a frown in a patient. This is really easy for us. So they've been wonderful in helping with some of these analyses. And I just want to give a shout out to our funding. Some of the funding started from AB Nexus back in 2020 that helped connect Rob McCurdy, Jeff Sansbury, and develop some materials. The Gates, of course, Frontier Fund, and you want to talk about T32?
I'm currently funded by a T32 as well.